Okay, this is the final prep for lecture 10 for PCR. And it really is about being able to trust the results we have, to be able to trust that the interpretation we're making, especially with diagnostics and that sort of thing, being able to trust that the interpretation we're making is correct. And that's because that's by doing the appropriate controls. And so PCR is a very sensitive technique. As I mentioned before, if you have even a single piece of DNA in a tube, you should be able to amplify a product. And so what that means is your mistakes can be amplified along with the intended template. And so it's important to be very careful when doing PCR. For example, most labs have what we call now barrier tips. They're tips that have a little barrier, like a cotton plug or a fiber plug at least, so that if you make a mistake pipetting, you won't get DNA. You'll get it into your tip maybe, but it won't contaminate the actual pipetter that you have. And there are different ways of trying to ensure that we're very careful, you know, the way we do the pipetting, the order in which we do the pipetting, you know, using obviously sterile equipment and so on that's been autoclaved and all that kind of thing. So being very careful is really important. But you also, it's easy to, for example, if I am doing work with human primers to human genes, there's a real concern about contaminating a sample because it only requires the tiniest flake of skin or something. If you talk and you make a mistake and a little bit of spit comes out, if that goes in the tube, you've added some DNA in there. And so we want to be very careful. We want to be sure that what we're amplifying is what we think we're amplifying, right? And especially for diagnostics. If I'm testing food for the presence of bacterial contaminants, I need to know that when I see a band, it really means that the food was contaminated, because somebody's going to be throwing that stuff away, and there will be a consequence to that. And especially I want to make sure that the absence of that product is meaningful. That is, I'm not telling the company, oh, don't worry, there's no bacteria in your food, and it turns out there is, and because of mistakes, my results were not correct. So if we see a band, is it because we used the right template and we amplified the template we think we did, or is it because one of the reagents had some template DNA in it? If we don't see a band, is it because there is no template for that particular pair of primers? For example, the food that is not contaminated with bacteria won't amplify a bacteria-specific gene. Or could I have made a mistake? Maybe I forgot to put something in the PCR reaction. Maybe I used the wrong temperature or something like that. So the idea is to guard against any mistakes and to be sure that when you're interpreting, you're making the correct interpretation. Okay, so for negative control, so we always perform a no template control. A no template control is one where you put in the buffer, the primers, nucleotides, enzyme, and so on, but there's no template added to it. So if you don't give it any DNA to copy, it can't produce any DNA. So we expect when we run a gel to see no band in there. And if there is a band in there, then somehow we've contaminated one of the reagents. We haven't been careful with our pipetting. And not just any DNA is contaminating. It's DNA that is complementary to the primer. So if we use primers specific for P-element sequences that we find in just a few fly species, that's not going to, if I drop some of my own DNA in the tube, that's sloppy, and that shouldn't, you have to be careful, but that's not going to be a problem for the PCR, because primers for P-element DNA are not going to recognize my human DNA. So it's not just contamination. That's what most students will say, contamination, but it isn't just that. It's DNA contamination, and it's not just DNA contamination. It's a specific kind of DNA contamination, one that can act as a template for the particular primers. So this is an example. Never mind about this because we haven't really talked about it yet. But so you've got basically 4 people that were tested, say for coronavirus or something like that, and are negative control. And so when we look at this result, we're thinking, oh man, these guys, they all have the virus. But the question is whether they do or don't. And the answer is we don't know. We can't say because the negative control has a band in it. Maybe the person doing the test, running the PCRs, is infected and is contaminated, or one of these DNA samples had the virus and the others didn't, and somehow we ended up mixing them up and not pipetting correctly. There are many places where mistakes can happen, but somehow this tells us we cannot trust these results. So I look at this and I go, well, I can't really tell you if, we're going to have to redo it and find out for sure. Okay. Now, if we don't see a band in a gel, we assume that the template didn't have the sequences. So we would, if we saw no people generating bands, we'd go, oh, that's great. None of them has the virus. But what if I made a mistake? What if I added one primer twice and the other one I missed out? Or I just, I grabbed the wrong buffer, or I didn't put the enzyme in the reactions and so on. We always include a positive control. And so that's the one where we use the same, when we do these reactions, we make a big mix and then we divide it up. So we put all the buffer and the enzymes and nucleotides and stuff together, and then we aliquot them into separate tubes and add the templates separately. So in the positive control, we would add a known template, and we would use those reagents and the proper conditions and so on that we know have worked before, so that we're sure that if we don't see a band, it's not because we made a mistake in setting up the PCR, it really is a negative result. And so here's an example. So this looks really good. So I'm seeing no bands here. I don't expect a band here. But what about this? So this is our positive control. This is the one where we should see a band, but we're actually not seeing it. So again, this is a case where this is most important for diagnostics. For some things, the positive control is not as important, but in cases of diagnostics in particular, it really matters. Okay, and in this case, most likely a pipetting error was made. We can't trust these results either. So we still cannot tell the poor people being tested whether they have the virus or not. Okay, so here's a different result. And I'm looking at this and I'm going, okay, number 1 and number 4. So first of all, I'm not seeing the same result in everybody, so that's a good sign. I'm seeing no band here and I'm seeing a band here, which is exactly what I expect. So this makes me happy because now I can go, being happy is the most important thing, of course. Now I can go, okay, person number 1 and person number 4 do have the virus, and I can reassure person number 2 and 3 that they don't, right? Oops. And so, yes, we can trust these results. So you will be, when you do your dog project, we will have both negative and positive controls. And we will use the no template control where we don't add any template, and then we'll use some DNA of dogs that have been used in the past which have worked very consistently. And so you'll get a chance to see how this works later in the semester, but in the meantime, understanding these controls is really helpful.